Okay, happy Friday. We're just going to make a start at this time, and if we have more attendees, then we'll update as they join the meeting. As always, these live online sessions are an additional tool, so not a required part of your course, but I do think that they add an element of more of an interaction and more of a chance for us to uh, address things that come up as we go through the week of learning together. So this week on this course, we have been looking at tissues. So as usual, I want to offer this live online session, first of all, as a chance to kind of highlight the nutshell version of the learning material for this week. But in addition to that, I do want to spend a little bit of time talking about just general updates on themes that have come up either in my one-to-one discussions with students or on the discussion forums or in other ways. So I think it's always a good chance to kind of, in addition to announcements, I want to provide a space where we can connect and make sure that if there's anything else that I can help everyone to be successful on this course, that I am able to deliver that. It's also a chance for me to hear feedback from you. So if you're not able to make one of these live online sessions, please never hesitate to share your ideas, thoughts, what can be done differently, or what we should keep doing, what's working well. So normally I would spend a little bit of time doing a check-in, seeing how everyone who's present is doing, but I think that today we're just gonna jump past that and start with the very first things that I had on the agenda, which is to talk a little bit more about the what's going on in terms of the um, items that I wanted to update you. And we're going to start on talking about the discussion forums. So let me again share my screen. This time we're going to look at our Canvas course. So I've done the settings so that it looks like I would be a student on this course. My instructor view is usually a little bit busier, but this should be very close to what you're used to seeing when you come to the course. And one of the things that I think I did a little bit of changes, and for that reason, I want to make sure that I address those, is that I had those discussion forums for us as a space to have discussions, dialogue, share resources, clarify more challenging concepts of the weekly material. And because it's important for me to also provide multiple ways for you to earn points on this course. And I wanted to add an incentive for you to take part on this discussion forum. So you'll notice that they now do have a due date, and they also have points associated with them. But notice that discussion forums are worth of 0 points, so they're not required. Since I did not require them at the beginning of the course, I'm not suddenly going to change the requirements. They're an optional place, optional resource, an optional activity that you can take part. But I do want to recognize anyone who takes part on these activities. And for that reason, I needed to associate as points value for the chapter discussions. I do realize that it's a little irritating for some students that you will now also see it listed as a due date activity. But remember, if it's worth of 0 points, there's no requirement to do it. Rather, I appreciate anyone who wants to do it, and I will give extra credit points for those who participate. And we have had some great discussions on these discussion forums, so feel free to jump in there, answer. I usually send a lot of prompting questions. Often, even when I get a response, I then expand on that topic and might have follow-up questions. I've seen some of the students sharing most amazing resources. So if you find something that has been helpful for you, I can guarantee that other students will find it helpful as well. It's a space also for you to ask questions. And again, I encourage you that if you have questions, that you also then consider of whether it would be helpful for you to have a peer who answers to that question. So feel free to jump in if you know an answer to something that's being asked. It doesn't have to be me who provides the answer. Oftentimes, a student is more successful in explaining a challenging concept to another student than an instructor. So I wanna promote that. So that's the first thing that I wanted to say about the discussion. So not to worry about them having now a due date and having them a points value associated with them. They are definitely still an additional resource, not a required resource. The other thing that I wanted to talk a little bit about is these additional optional study materials that I have at the end of each learning module. A few of the students have really taken advantage of these, and that's amazing. So even though I mainly want you to do the homework assignments, and for most weeks we have a quiz and practice anatomy, and we might have simulations, we might have short essay questions in addition, but what I really ask you to do are these homework assignments. If you feel that these homework assignments, you did them and they were rather easy, or you would want to apply the content from those or move the content from those to more clinical scenarios, these additional optional study materials, which I label as want more question mark areas, are for you. So in these want more areas, what you will see, we have a bunch of additional material. There's a short synopsis of what I want us to talk about that week, key points listed in a bullet point manner, and also I have various prompts. You might want to answer to all of those discussion and application and clinical questions, or you might want to answer to only some of those. It is completely up to you. I also provide a bunch of reading resources for you. Like I said, I also have critical thinking questions and clinical application questions. Again, you are free to choose how much of these you want to answer. I get some students who answer each and every single one of these. I get some students who pick one that seems interesting to them and answer to that. Or some students who find one particularly smooth or easy to answer and they tackle that. Sometimes students ask me that, well, what's expected? How much should I do of this? Again, these are an additional resource, so they're not required, but to give you an idea of the points value associated with these want more questions, my kind of rule of thumb that I've been following has been that if you were to answer to all of the questions, so it would include the discussions, critical thinking and clinical application, and you answered all of those correctly and you answered kind of in a sufficient detail, you do not need to write full essays, but more than just a yes or no, a little bit of more than that. If you did all of that, the rule of thumb that I've been providing for students is that completing these would be worth of the same number of points as we have available on the homework assignments that I require from you. So typically it's about 20 points worth of extra credit that you can earn by doing these. So again, remember, those are not required. They're an additional resource for those who either want more of a challenge or want to tackle homework in a different way. I'm very big on giving multiple ways of collecting those points. So however you want to answer to those questions, you could do traditional paper notes, you could write your answer on a computer, or whatever you wish to do. Once you have worked out answers to those questions, you would upload your submission to the second space under the heading Want More. And again, notice it's worth of 0 points, so it's not a required activity, but if you do it, you can earn extra credit. And you can submit your work in quite a few different ways for these Want More assignments as well. You could, for example, choose to simply load a file. It could be that. let's see, we could, for example, upload a Word document, or we could upload a picture file of the notes that you've made, whatever you choose to do. What's important for me is that it is a file that I can open in Windows compatible environment. So sometimes I know that we have students who work on Mac environment, on Apple hardware, software, whether it's a computer, whether it's a mobile phone. Unfortunately, our grading system will not open those files, so you would have to submit your work in a format that is compatible with the Windows environment. So you can submit it there and then just send it in to me as a file. You do have other options as well. If you preferred, you could copy paste your test answer there, and again, submit your work. I'm a little hesitant with students who want to do file sharing. You can completely do that, but they do tend to have often hiccups on the process. If you do file sharing, often those requirements of who's allowed to access the file, view the file, edit the file, comment on the file, ends up being that then I send you messages back and forth that I see you did the work, but I can't open it. So I really encourage you either to do the picture version or file version, or copy pasting your answers. And like I said, then you are able to collect those extra points. So those were the two of the biggest things that I wanted to talk about in terms of questions that have came up from students. And you'll actually see that I posted during the midweek a little bit more detail about these both, so discussion forums and want more assignments, as our announcement as well, with specifics of where to access questions and where to upload your answers. So that's something that might be helpful for you. I also received a very good question from a student who was interested of whether there's any tips or hints that I could do or provide in terms of how to succeed in this class. And again, if you go to the Start Here module, you'll be able to see that I have there a page how to get a good grade on the course, and I provide some pointers. In addition, on this page, in response to this request, I use the materials that I use in many of my other courses. We talk a little bit about studying in general, but especially studying anatomy and physiology. We have a look of different learning styles, how they translate to study of anatomy and physiology, and also talk about case studies. So hopefully that will be a useful resource for those who want to have a little more dialogue on that. If that is you, and if you do wish to have a session with me in terms of one-on-one, remember I offer office hours, quite a few options. I have office hours on Mondays, on Wednesdays, and then during Fridays I have on a per appointment office hours. But I can guarantee you that if you have an interest to meet with me and you reach out to me, I will be there to work with you to find a day and time that works, even if it's outside of the office hours. That's something that I really much want to facilitate, that you're able to get hold of me anytime that you wish. So those were the big kind of pointers or important things that I wanted to discuss in terms of the updates that have came up in our discussion. And having said all that, I think that we are ready to move on to have a look of this week's learning materials and what kind of key points we might see from those. So kind of a mini lecture. Obviously, you will have a full-sized lecture of everything in your Canvas course. So this is highlighting just the main points for our chapter material. So let's do that together and have a look of what we can find and what were the most important things to take away from this week's learning material. So where I want to start is taking us back to what we saw, that the study of life, which is essentially biology, can be done in many different levels of organization. So we can look at the smallest of things. We can look at elements. So these are the basic components that all things are made of. You can't really break down elements further with traditional chemical means without putting great effort to that. Obviously, we talked about the atoms. We do know that atoms have subatomic particles, but again, in traditional chemical means in a lab setting, atoms are as small as we go. We saw that when we have two or more atoms that join together, that forms a molecule. And then we had some macromolecules, which were particularly large molecules, kind of essential for life. And we'll get to discuss those more later on our A&P studies. For example, when we talk about nutrition and metabolism, we'll look back to these macromolecules. We also saw that a cell was the smallest basic unit of life. We saw that cells were made of cell organelles, but really we can study the life as a whole, as a one unit in a cell level. So what we end up seeing is that we have some simple cell organelles, so organelles that are nothing more than just a single cell. But we're also going to have multicellular organisms such as us. When we study humans, obviously we are made of many cells, and many cells that work in different manners to accomplish the whole goal of producing the human organism. So when we have cells that come together, and these cells perform a similar function, we're talking about these being tissues. And in organs, we saw that we had multiple different tissue types on a same area performing a similar goal in terms of working together, each tissue type contributing their own element, but this formed an organ. And then we can have multiple organs that all work together to perform a bigger of a function together. I can think of, for example, that being things like a digestive system. A digestive system is made of many organs, but as a whole, the goal is shared between all of these. We also saw that these cells that we were talking about are rather small. So most cells, you would need an additional method to be able to study them and see more of them. And this is where really the microscopy is going to step in terms of the scale of the matter that we're studying. So in classroom setting, I'm sure many of you have used light microscopes. Light microscopes are good, relatively affordable option to see structures that are too small for the naked eye to see. I would say that on average, we will see the cell, we will see the cell membranes, boundaries, cytoplasm inside, and nucleus, but that's about all we can see with the light microscope. To see the smaller cell organelles, we typically do need electron microscopy techniques to help us with that. So unfortunately, we do have less availability for electron microscopes. These are expensive apparatus, and not every institution has those available, at least for all of the students to access. But those have done a significant contribution to our study of cell organelles. So a couple of terms that I wanted to clarify here. So if we're studying cells, we're talking about the field of cytology. When we're studying histology, we're studying tissues. And remember, tissues were when we had multiple cells coming together to form these larger masses of cells that shared a function together. So tissues are groups of similar cells, and what we end up seeing that these cells share a similar structure. They usually often have a common or related function, and they are often located in same area to form one tissue, but we will see tissues all over the body, and even though they're located at the different parts, the same tissues do typically share very similar structure and common function. Again, the term that I'm highlighting here, the term histology, which referred to the study of tissues. So even though there are many different tissue types, we can classify the basic tissue types into four main groups that are the four main tissues that I want you to be aware of. So we'll talk a little bit about epithelial tissues, connective tissues, muscle tissues, and nervous tissues. And I'm going to heavily emphasize today on our meeting, and I think a lot in this chapter as well, the role of epithelial tissues, because we don't really get to talk about these tissue types anywhere else. Other tissue types, we'll talk them in more detail once we get to those chapters. But epithelial tissues really are kind of leading us well to our first organ system that we'll have a look of, which is going to be the integumentary system, so the skin and associated structures. So I feel that that's a great place for us to start. If we look at epithelial tissues, like I mentioned, skin would be a good example. We also see epithelial tissues, for example, on the digestive tract lining. And these form boundaries or protective layers and also facilitate secretion, absorption, and filtering through these layers. So important tissue type that does an important role in our body. Talking really quickly, kind of introducing the connective tissue. Connective tissues really do what the name says on the tin, so they connect body parts. These could be bones, tendons between muscles and bone, ligaments, they could be fat and soft tissues, or they even could be blood. And a lot of time I think that that kind of a physical connection between structures is obvious for us, but you can think of blood, for example. It connects different body parts by transporting materials, so it very much falls under this category of connective tissues. So whether it's physical or physiological connection that we're seeing, that's up for grabs. So their function is really to support, protect, bind. There's a range of functions that connective tissues do, so very varied group. In terms of the muscle tissues, there's really going to be three main groups of muscle tissues: are skeletal muscles, cardiac muscles, and muscle cells, and smooth muscle cells or tissues. And skeletal muscles are really these big muscles that we think about in terms of moving the body, connecting body parts. We'll study those on the surface of the body, and they are under voluntary control. So you get to choose what muscles you move. And in fact, we often see them in pairs, that there's opposing movements we can do with opposing skeletal muscle structures. In terms of cardiac tissue, we find that only in the heart, so important for pumping the blood around the cardiovascular system, around the body, under involuntary control. So you don't really have to think about, I'm going to have a heartbeat now. It just automatically happens. And finally, smooth muscle, which is going to be important when we talk about, for example, regulation of the vessel diameter in blood vessels, or we see smooth muscle also in the digestive system, for example, also forming sphincters and so on, controlling the movement of material through it. So muscle in general, you could say that the biggest function, I guess, or the most obvious function is that as muscle contracts, it generates movement. But we'll again end up seeing that muscles can do a lot more. We, for example, use muscles when we are regulating body temperature, when we're getting too cold, so we can start to shiver to produce heat and so on. And finally, our nervous tissue. I wanted to mention that as an example of a tissue type that's responsible for internal communication within our body. So this could include our brain, our spinal cord, our nerves. We have information being collected from different parts of the body, or even external to the body, carried to the central nervous system, where it's processed and then commands passed out from the central nervous system to other parts of the body to perform activities. So that's our nervous system. Let's dive into each of these main tissue groups in a little bit more detail, and we're going to start by looking at epithelial tissues. So the term that you sometimes see, epithelium, referring to that. And there's really two main types of epithelial tissue that we're going to see. We're going to have epithelial tissues that form coverings and linings of the body. And again, skin would be a great example of that. That's an external surface where we find this lining of epithelial tissue. But we have many internal surfaces where we find linings as well. And in addition to that, we had glandular epithelia. So these are the glands. Salivary glands are one example that I highlighted there, but sweat glands, oil glands on the skin would be another good example. So these are responsible for being secretory tissues. They produce secretions. So again, at large, if we think of the epithelial tissues and their functions, a large range of functions: protection, absorption, filtration, excretion, secretion, sensory reception, and so on and so on. Here I have a rather interesting and important table for us to look at. And the reason why this table is so valuable for us is that it actually gives us two ways of classifying epithelial tissues. Not all epithelial tissues are the same. And let's see if I can take out a laser pointer for us. So we're going to start with what we're seeing here on the left-hand side of classifying epithelial tissues based on the number of layers of the cells that we're going to see. So simple epithelial tissues are made of just one single layer of cells. So we see three different examples. All of them are characterized that we have just one layer of cells that's resting on the basement membrane. Stratified epithelia instead is made of multiple layers of cell. And these multiple layers of cell again rest on basement membrane. So stratified referring to two or more layers of cells that form an epithelial tissue. In addition to this, looking at how many layers of cells we have, we can also look at the shape of the cells. We see here squamous epithelial cells are kind of these flattened disc-shaped cells. So if we put the two together here, we're seeing simple squamous epithelia. We could also talk about stratified squamous epithelia, as we're seeing here. So again, we're putting together the shape and the number of cell layers. When we're naming these multi-layered, these stratified epithelial tissues, we're looking at the outermost free membrane, layer of cells and their shape when we're doing that definition. In addition, in terms of the cell shape classification, we also have cuboidal epithelial cells. These are cube or dice-shaped cells. So again, we're seeing simple cuboidal epithelium, one layer of these cube-shaped cells, or stratified cuboidal epithelia, where we would see multiple layers, and these cells at the outer free layer, are gonna be shaped as cubes. And finally, we have columnar epithelia. Columnar epithelia look like pillars or columns, if you wish, and we see that we can again classify them based on the number of layers, so simple columnar epithelia here, or stratified columnar epithelia. One thing to notice about these simple columnar epithelia cells is that sometimes they have these little hair-like projections also on the surface. So these cilia can play an important role in moving materials along this layer formed by epithelial tissues, free membrane. So we see this, for example, in uterine tube, where these cilia sweep the egg cell towards the uterus. We see these cilia in respiratory system, where these epithelia sweep the material from the lower parts of the respiratory system to the upper part, so we can get rid of that material, and so on. So this classification is worthwhile. It's going to come helpful for us as we look at, for example, the integumentary system, the skin later on. We would see that skin is very much made of largely of stratified squamous epithelia. Another term that I wanted to introduce here is going to be pseudostratified tissues. So you remember that the word stratified meant multiple layers, but in this case, we're actually seeing pseudostratified the epithelial tissues. Remember, the word pseudo refers to fake, if you wish. So we have, for example, pseudosciences. These are things that some people claim that is scientific, but it's really not, such as reading tea leaves or astrology or so on. So pseudostratified means that we have cells that might appear as if they were multi-layered, but in reality, each and every one of these cells is just one layer. They all connect to the basement membrane. So it looks like multiple layers, but in reality, it's a single layer of cells. Now let's finish off our epithelial discussion by looking at the glandular epithelia. So glands are cells that make and secrete fluids, and glands may be formed by single cell or multiple cells. We can classify glands in two groups. We can classify glands as endocrine glands. These are glands that produce hormones, and these hormones are released to the bloodstream where they travel to all parts of the body. Or we can classify them as exocrine glands, which is what I think my students more often think about when we're talking about the glands. So what's characteristic for these exocrine glands is that we have the gland part here, and that gland is responsible for producing that secretion. But in addition, from the gland, we have a duct that comes out. And this duct can be longer or it can be shorter. But what's important to notice is that this duct carries the secretions from that gland. So the ducts are important in that sense. And these ducts open up very much locally where we have that gland located. So the secretions from that gland don't travel a long distance, rather they tend to be fairly localized secretions. So here, for example, we could have tissue. Let's draw a couple of epithelial cells, and then we see this gland embedded in there. So we're seeing that the gland's duct opens where we would have the secretions being released to the surface. And again, let me tidy up a little bit this picture since my computer was too eager to take out. So if we have the gland, and the gland was producing a secretion, let's say that there's the secretions that it's producing, it's going to release these secretions very much locally on the surface or on the structure where that duct opens. So again, I think a sweat gland is a particularly good one that we'll be talking about later on. The sweat glands are located within the skin, and then they release the secretions on that area on the surface. And these exocrine glands are what we will be focusing on. We'll leave the endocrine glands for later chapters. I think you'll start your BIO202 by talking about hormonal systems, so we'll leave them for that. Our main focus on this course will be on exocrine glands. Like I said, these glands can also be classified based on, do we have one cell that makes the gland, or is the gland a creation of multiple cells that come together to form that gland? Let's move on from the epithelial tissues to talk about connective tissues. Connective tissues form a huge range of functions. Like I said, my rule of thumb would be to think of them as connecting structures in the body, connecting parts within the body. So they can serve purposes in binding and supporting body parts, protecting, insulating, storing reserve materials like fuel or calcium in the bones. And they also play a role in transporting substances, and that's where the blood was a good example of a special type of connective tissue. There's really four main classes of connective tissues that I want to talk about: connective tissue proper, cartilage, bone, and blood. And if we are thinking about this, I think that we can kind of think of, for some of the examples where we would see connective tissue proper, for example, in the lungs or fat cells, tendons, ligaments. And these include special groups of connective tissues, such as loose connective tissue or that connective tissue. Cartilages instead, like I said, tendons, ligaments would be good examples of those. We see hyaline cartilages, we see elastic cartilages, and you can read a lot more about those in your textbook. When it comes to the bone, bone again can be classified further, as we'll see in our subsequent chapters, either as a compact bone or spongy bone. So some considerations there as well. And like I said, the blood is an interesting connective tissue since it connects body parts more of through the transport functions. But blood, again, on your BIO202 courses, you'll study them blood in more detail. We'll see that blood is made of various different cell types, red blood cells, white blood cells. We'll see plasma, we'll see platelets, and so on. So very varied group when we talk about connective tissues. All connective tissues do share some basic elements. So connective tissues do have the ground substance, they have fibers, and they have cells. So that's kind of a good way to start to break apart these different connective tissue types, thinking what is the ground substance, what are the fibers, what are the cells that we're seeing on this particular connective tissue type. And let's move on to talk about muscle tissues. Like I mentioned earlier on, muscle tissues can be considered as if their main function is the generation of the movement. Obviously, there's more to the story than just that, but true contracting muscles do create movements, and those are what we most often end up seeing. Like I mentioned, there were three types of muscle tissues: skeletal muscles, cardiac muscles, and smooth muscles. And I believe that next I have a review of each of these, and we'll have a look of an individual cell of these different types of muscle tissues. And remember, there would be lots of these cells forming then the tissue. So let's start with the skeletal muscles. Skeletal muscles are these cylindrical shaped cells. They have multiple nuclei, so multinucleated cells, and they are also striated. So we have these stripes running across. Skeletal muscles are under voluntary control, so you get to choose that now I'm moving a muscle, now I'm moving a different muscle. So you do have a voluntary control over those. Sometimes students get confused. They ask, how can a skeletal muscle have multiple nuclei? I thought a cell only had one nucleus per cell. Well, what we're going to see is that actually skeletal muscles form during the process by fusion of multiple cells, so you are left with multiple nuclei as a result. Let's talk a little bit about cardiac muscle and cardiac muscle tissue or cardiac muscle cells. These are often cells that are very branched, and since they are branched, this allows us to have multiple cardiac muscle cells connected to other cardiac muscle cells, which is really important for the main function that we have for the cardiac muscle. In these points where the cardiac muscle cells connect to other cardiac muscle cells, we do have a special adaptation known as intercalated discs. These intercalated discs allow easy movement of materials and messages across it from one cell to another, and this is again important for coordinating the functions of the cardiac muscle cells, so we get coordinated contractions, so we can have all parts of that tissue within that area of the heart contracting at once, so we get the functioning of the pump-like pumping of the contraction of the heart, so we'll get the blood moving. If each cell was doing their own thing, we wouldn't get that coordinated movement. In addition, we see that cardiac muscle cells are described as being uninucleated, so one nucleus, usually fairly central, and we'll see striations in those as well. The thing to notice about cardiac muscle cells is that they are involuntary. You do not really have a control over your heart rate or heartbeats. That happens automatically. So that's our cardiac muscle cells. And let's finish off by looking at the smooth muscle cells. Smooth muscle cells are often spindle shaped, but this shape can change a lot depending on how the muscle actually contracts. They do have one central nucleus. They do not have any striations, so the cytoplasm is clear. And again, these are under involuntary control. Remember, we saw smooth muscles for example, regulating the vessel diameter within the blood vessels or blood vessel wall, or for example, within the digestive system. So that's our muscle tissues, and let's finish off by having a look of nervous tissue. Nervous tissue, the main function is to regulate and control body functions, so collecting signals, bringing them to central nervous system, processing those signals, and passing messages from central nervous system to other parts of the body to trigger responses. We do see that the main components of the nervous system are going to be our brain, our spinal cord, and then other nerves that we see. We're going to be actually spending a rather long time talking about the nervous tissue later on this course when we get to the final bigger chunk of chapters where we talk about the nervous system. At this point, I do though want to mention that we have two types of nerve cells. So we have neurons. So these are the nerve cells themselves that did this function of passing messages, sometimes processing messages. And then we have something known as glial cells. Neuroglia is responsible for kind of protecting, supporting the neurons themselves. So without neuroglia or glial cells, neurons often would struggle to be able to do their function. They protect them physically, but also physiologically, providing resources, clearing away waste products, and so on and so on. In fact, we've seen, and I think I mentioned this in our discussion forum on someone's post, that in fact, we initially assumed that Alzheimer's disease and figuring out what's causing it and then solution to that would be accomplished by studying just the neurons. But we are finding out that the glial cells are where the issue happens with Alzheimer's disease. So again, we should not ignore a cell type that we're not familiar. There might be huge insights that we get out of that. And this brings me to the end of everything that I was planning to tell in terms of these different tissue types as a revision type of an activity. I'm going to stop sharing my screen. And what I was thinking that we could do for the rest of our time together on this meeting is to have a look of, again, a couple of the examples of the homework questions that I'm asking you to do. So while on the background, when I'm not yet sharing the screen, I'm just navigating to our Canvas course. So give me a second to get there. I'm ready to share my screen with you. So let's have a quiz for this chapter 5. What we need to know about chapter 5 quiz. Well, there's going to be 12 questions. They're going to add up to 10 points. And let's have a look at a couple of examples of those questions. First question would be multiple choice. You're choosing which are the 4 primary tissue types in the body. And what we're going to see that they were, of course, are epithelial, muscular, connective, and nervous tissues. So rather easy, nice warm-up question on the quiz. We're being asked what is not a primary tissue type. So bone is a type of connective tissue, but bone itself is not the primary tissue. When we're talking or answering to a question, which of the following terms describes a thin slice of tissue mounted on a microscope slide, that would be a histological section. Remember, histology was study of tissues. And usually when we're studying tissues on a microscope, we want to take a thin cut of that tissue so we can then stain it and study it and understand it better. And you will also have some drag and drop questions. So here, if, for example, you would be asking to tell the statements, are they more of descriptive of muscular tissues, nervous tissues, epithelial tissues, or connective tissues? So then you take each of the statements and drag them where they most are appropriate. And I think your textbook, the lecture material that I provided on Canvas, all of that good stuff is going to be super helpful as you tackle these questions. Let's have a look at one more question from the quiz. Here we're being asked, how would you describe an epithelia consisting of single layer of cells in which all cells rest directly on the basement membrane and all cells reach also the apical surface, so that free surface. These were, of course, our simple epithelia. Simple meant one layer. Stratified would be 2 or more layers. So I think that hopefully helped you to again get a feel of what to expect in terms of the of our quizzes. And let me just jump real quickly to show you the practical as well, or practice anatomy questions. Let's get that working. I want to show an example from there. I think that at this point you should be able to see practice anatomy this time around is going to be 14 questions, 10 points in total. And again, here you get to see either diagrams or pictures and label different parts of the histological samples. We're seeing, for example, multiple different tissue types on this particular diagram. There's going to be all kinds of drop and drag and drop questions. You'll see those cilia that we talked about, basement membrane, and goblet cells, which are secretory cells. So good questions in that sense. I think that they're going to be rather useful for you to study. And the final thing that you will see on this week's assignments is going to be that simulation activity. And let me just pull that up for you as well. It is about histology, since we've talked about histology, what we would do in a lab with my students normally. And let me see, I don't think I'm able to share it with you, but it is a very fun activity, allows you to prepare the slide yourself as if you were in a lab setting. So you get a little bit of a flavor of that process as well. So what I'm gonna do, I'm gonna jump one more time to share the screen and then we'll call it a day. So like I said, this is all I have planned for us for today, but I think that this is going to be a fun week, a lot of revision of what you already know, and that gets us all set for success when we move on to our first actual body system in 2 weeks' time. Next week, we'll still have a look of a little bit of the basic concepts. We'll talk about, for example, cells. We'll talk about genetics and so on to refresh the basic concepts that you learned in your general bio classes like BIO201. So that wraps up all from me from today. I look forward to seeing you in discussion forums, interacting with you, and I think it's going to be a great week. As always, I'm here to help, so do not hesitate to message me. Until I see you at the other spaces, thank you for joining virtually, and bye.